Hello, I am Professor Sims, and this video is an introduction to compound light microscopy. This is the third in a series of 10 lab sessions held as part of my online Laboratory for the Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this lab include learning to identify the parts and the function of each part of a compound light microscope. You're going to learn to properly operate and care for a compound light microscope. You'll become competent in basic wet mount slide preparation. You're going to learn to observe various specimens using a light microscope and also using the Foldscope instrument. First, we'll talk about compound light microscopes. Microscopes in general are needed to view microorganisms such as bacterial cells, which are so small that they're measured in micrometers. A micrometer is 1 × 10⁻⁶ meters. Here you have a nice figure demonstrating the limits of what you can see with the human eye versus a light microscope or an electron microscope. The human eye, obviously you can see the human or hand or finger, but the limit here is going to be about the size or the thickness of a human hair. For things smaller than that, you're going to need some kind of microscope. A light microscope can see individual human cells, bacterial cells, some subcellular structures. And if you want to see something as small as a virus is actually 3 orders of magnitude smaller than a bacterial cell, and it's measured in nanometers. So for that, you're going to need an electron microscope. And an electron microscope, depending on which type you're using, and we talk about the types of electron microscopes much more in the lecture course, but some of them can see as small as a single atom. The compound light microscope, which sees within this range here, is generally what is used in a traditional microbiology lab course. Although you will not be working with a compound light microscope at home, you will still need to know the parts of the microscope, which are all defined here. Be sure you know the name and the location and the purpose of each of these parts. This figure here illustrates the parts of the scope. You have the ocular lens, objective lens. This is the nose piece that holds the objective lenses. The arm. You have coarse adjustment knob around the outside here, this big knob. And on the inside part is the fine adjustment knob. The base. The illuminator, which is the light source. The condenser piece, and then just below that is the iris diaphragm. This is the stage where you put your slides. These are the slide holders here. Again, just be sure that you can identify all of the parts and know how to properly use each part. As far as focusing and observing specimen, it is important to understand how the microscope works in order to be able to focus and view your specimen. So in a compound light microscope, you have light that comes from the source, the illuminator. It is first focused by the condenser, the lens in the condenser. Then it enters the objective lens, where it is magnified in order to produce a real image. So you start with a virtual image, and then you end up with a real image that you can view with the eye. The real image ends up being reversed, so it's mirrored upside down and it's mirrored horizontally, which becomes important to know when you're actually trying to move your slide around in order to get a better view. You have to understand that it is going to be the opposite, both vertically and horizontally, from how it looks like it is on the stage versus how it's going to look to you through the ocular lenses. Therefore, it's going to take a little bit of practice to orientate yourself when you're using the microscopes. And that applies to a compound light microscope as well as the Foldscopes, by the way. You need to know the steps involved in using the 10x and 40x objectives, and also later on we'll talk about the 100x oil immersion objective. So first you want to turn the illuminator on. You start at about half power because it's very bright when you first start on 10x. You use your coarse adjustment knob to lower the stage. You set the objective to 10x. You place the slide on the stage, and you use the coarse adjustment knob to raise the stage so that you have a minimum working distance. The working distance is just the distance between the objective lens and the slide that you're trying to view. You will adjust the interpupillary distance, the distance between your ocular lenses, and that helps to make sure that you're not seeing double And then you look through the ocular, rotate your coarse adjustment knob slowly so that you can get the specimen into view. And then once the specimen's in view, you use fine adjustment to focus the image further. And then you'll rotate the objective lens into the 40x position, and you'll refocus again using just the fine adjustment. With a light microscope, compound light microscope, you're only ever using the coarse adjustment knob on the 10x objective. Anything above that, you have to use only the fine adjustment. Mastering the light microscope instrument requires understanding and applying the concepts of microscopic field, parfocal distance, magnification, and resolution. The idea of microscopic field and parfocal distance is basically, as you go up in magnification, your specimen that you're looking at, it's going to look very different. And the reason for that is not only because it's getting bigger, but because you're looking at a much smaller portion of the specimen. So your microscopic field, when you're on 10x, as you can see, a much larger part of your specimen. But then when you go up to 40x, you're zoomed way in, and you can see a much smaller part of your specimen. When you're on 100x, it's even way smaller than that, okay? So these are all the same specimen, just at different magnifications. The parfocal distance concept means that you should be able to move from one objective to the next without doing very much adjustment of your specimen. In other words, you don't want to move it around too much, and even adjusting the fine adjustment knob should be very minimal. You want the changes that you make between the objectives to be very slight, very minimal changes. Because if you lose your specimen, you can't see it anymore on 40x, you're not going to find it again on 40x. You're going to have to go back down to 10x, start over, and then go back up to 40x. Same thing for 100x. So finding a bacterial cell in 10x is like finding a needle in a haystack. Finding a bacterial cell in 100x is going to be like finding a needle in a stack of needles. It's going to be very difficult. And that's because not only are your cells getting magnified, but so is all of the white space around it. And if you move this slide a little bit too much to where you're focusing on the white space, all you're going to see is white in 40x. All you're going to see is the white space in 100x. So that's the idea of parfocal distance. As for magnification, you need to understand that magnification and resolution are different things, but they do work in conjunction. So your magnification is measured by the ocular lens magnification multiplied by the objective lens. So magnification, total magnification equals ocular times objective. Most ocular lenses are going to be 10x. The ones I use in my on-site lab are all going to be 10x. And the objective lenses are the ones that change. So those are the ones that are on the nose piece and you can rotate. Some scopes have them as low as 4x. For microbiology labs, I generally have students start at 10x. And then there's also 40x and 100x objectives. So if you're looking through a microscope that has the objective set to 40x and the ocular is 10x, then that means the total magnification is 400x. And magnification really is just making something bigger. If you want something to be focused, well, that is actually resolution. Resolution is the ability to distinguish between 2 points, a specified distance apart, but far enough for light to pass between them. So basically what that means is you can have something that is magnified very nicely, but it is blurry or it's out of focus, or you really can't see the structures. All you can see is the color or the basic shape. In order for it to be clear and focused, you need to have good resolution. Resolution is determined by the numerical aperture of the objective lens that you're viewing your specimen through. The numerical aperture is a measure of the cone of light that can be gathered by the objective, and by using immersion oil, you can change the numerical aperture, so we'll talk some more about that in just a bit. But essentially all you really need to understand with this, you don't need to understand the physics of this, but you do need to know that the different objective lenses do have different numerical apertures, and that is what determines the resolution power of those lenses. For experiment 1, all you're going to need is a computer or another internet connected device. You're going to navigate to the following URL and you're going to complete a tutorial. It's a tutorial where it's going through the basic concepts of light, how microscopes work, how different types of microscopes work. So not only the light microscope, but phase contrast, fluorescence. I think it even dives a little bit into electron microscopes. So you want to click through all of the links or all of the steps there and watch all of the animations. Record your answers to the guided questions and the lab report 2 template. Also, here's just a little short clip of what this tutorial is going to look like for you.
Light is created when electrons within the atoms become excited by absorbing extra energy from their surroundings. The extra energy causes the electron to move up to a higher energy level within the orbital shells of the atom. Since atoms seek stability, the electron will return to its original orbit, releasing a particle or photon of energy in the process. A ray of light is a steady stream of photons traveling along in an electromagnetic wave. The discovery of the law of refraction in 1621 made possible the development of the compound microscope. The basic compound microscope consisted of 2 convex lenses in series, an objective lens close to the specimen, and an eyepiece lens near the observer's eye. Light microscopes may illuminate the sample from the opposite side of the lenses, known as transmitted light, or illuminate the sample from the same side as the image-forming lenses, known as reflected light. Modern contrast in biological tissue sections can be enhanced by staining the sample. The most common stain is commonly known as H&E and is made from hematoxylin and eosin. Hematoxylin will bind to nucleic acid and will highlight the cell nucleus. Fluorescence microscopy is a common method of visualizing the structure of a cell. The specimen can either autofluoresce or be treated with a chemical.
Next, we talk about the oil immersion objective, the 100x objective. So the key here is you're dealing with refraction of light. So you should have seen in the tutorial how important it is to gather light and how important it is to try to keep your light from getting refracted away when you're focusing. And the refractive index of your slides is about 1.56, whereas the refractive index of air is 1.0. So what happens here is once you get down to the higher objectives, especially the 100x objective, you need to really capture all of your light, but you do have light that gets refracted in air. So in order to try to help with that, when you're using the 100x objective and you're in a really high magnification, remember 100x objective lens, if you're coupling that with a 10x ocular lens, that is 1,000 times actual size. So you're getting into really high magnification, you need to really focus with all of the light that you have available. So what we do with a 100x objective is we use what is known as immersion oil. The immersion oil forms like a little bridge between the light source and the slide and the objective lens. And that way you reduce the amount of light that is lost. It's very important whenever you're using the oil immersion lens that you clean the oil after you use it. Don't want to leave it on the lenses. And you're never using immersion oil with any of the other objectives, just the 100x objective. So again, here's some special precautions that you are going to be using for the 100x objective. Again, now this is for the on-site labs, but you do need to know how to use all of the parts of the light microscope and how to care for the scope, even for the online labs. So make sure you're never using coarse adjustment with the 100x objective. You only use the coarse adjustment with the 10x objective. You add oil to the slide by rotating the nose piece halfway between the 40 and 100x objective. Then after you've added the oil, you'll rotate the 100x lens into place. The lens should be touching the oil. So the oil is touching both the lens and the slide. So since you can't do this on site, you will be doing a simulation. For experiment 2, you're going to again need a computer or another internet connected device. You're going to navigate to this site here. You're going to complete the simulation and record all of your answers to the guided questions in the Report 2 template. This one is a pretty fun simulation. Here is a clip kind of demonstrating how to navigate this simulation.
Hello, Lab Assistant. Today, we'll view the gram-stained yogurt sample with a microscope to get an idea of the kind of bacteria contaminating it. To learn more about the purpose and techniques of gram staining, please take a look at the Gram Staining Virtual Lab. A short background about gram staining. Hans Christian Gram, a Danish scientist, devised this technique to let bacteria. We will use a light microscope with 10 times, 40 times, and 100 times lenses. The 100 times lens is an oil immersion lens. Basic lab safety procedures. Before you start, this microscope works by magnifying the sample on the slide using a series of lenses. Mouse over the different parts of the lenses are rotated on the nosepiece. View the magnified sample through the eyepiece. The arm supports the stage clip, the slide, the fine focus, the coarse focus adjustment, the diaphragm, the light source projects up through the slide to illuminate the sample. Start by making sure the microscope is plugged in. Take a closer look at the nosepiece. The number on the side. Rotate the nosepiece to the lowest magnification, the 10 times objective. Multiply the number on the objective lens by 10, the magnification of the eyepiece lens, to find the total magnification. For example, the 10 times objective would give you 10 times 10, or 100 times magnification.
Now finally, we'll get into some of the hands-on at-home stuff that you actually get to do, and it's not just a computer simulation or a tutorial. So for your online labs, you're going to be using a Foldscope. A Foldscope is an inexpensive yet very useful handheld paper microscope. It comes with a high-power lens and a magnetic coupler that you use to attach it to your cell phone camera. And if this Foldscope was designed to, quote, bring microscopy to the world. So the designer of these things, they are bringing them to schools that are underprivileged and that could never afford microscopes, and really just kind of opening up a world of discovery for children all over the world. It's great. Now for our purposes, we're using the Foldscopes because I couldn't send you guys home with $2,000 compound light microscopes. But this is a good alternative. It has a magnification lens. So the objective lens on these things is 140x. And then the resolution power is pretty good. It's 2 microns. So with those capabilities, you can view things within the range of bacterial cells, blood cells, other single-celled organisms. And of course, you can also see larger things like insects, algae, organic tissues. You guys are going to be keeping these things, and I've sent way more paper slides than you actually need for this class, so you can play around with these things and look at nature and that kind of stuff if you want to. You're going to want to follow the instructions that are within your kit for assembling the Foldscope, for creating slides, inserting slides into the scope, how to view the slides. The procedure for Lab 3 contains links to videos that will help demonstrate those instructions a little further. And I've actually included here just a little compilation of short clips from these videos. They're much longer and much more detailed, but here are just a few clips.
Let's practice preparing your own slides. First, I'm going to show you how to prepare one with a glass slide, and then I will also show you how to prepare your paper slides. I have here some water and algae sample that I took when I was going out with my friends at a picnic. So let me just take a little bit of sample here. Just place it on top of the glass slide. Then your kit comes with a cover slip stickers. So just take one of this. And then make sure you're holding the edges so that you don't leave your fingerprints right in the middle. And place it right on top of your specimen, like so. And it should stick pretty firmly, like so. If you're using paper slides, you're going to take one paper slide, put one sticker on this side. Make sure you're attaching it. And then flip it over so that this side is sticky. Again, place some samples that you have. And this could be anything from leaves to some pond water sample. Just a tiny bit will give you a magnificent world to explore. Like that. And then put another sticker over so that you're sandwiching your specimen with these stickers. Like this. And it's okay if something spills out. You can wipe it away. And if you're using paper slice, make sure that you put 2 blank paper slice underneath so that the thickness is just like the glass slide. Now let's practice preparing a sample with a microwell paper slice. These are great for pond water samples, which I have here. And if there are any swimmers in here, they get trapped in these tiny wells so that you can see them clearly. The first step is to put a strip of cellophane tape. You can cut it with the scissors like that. And put it over the microwells like this. And make sure that it's attached to one side. Now flip it over so that the sticky side of the tape is facing up. And you can take a little bit of water sample with a dropper like this. And then place them just a tiny bit. Tiny bit goes a long way. And then place it on top so that you're sandwiching the water sample with 2 pieces of tape. I'm going to flip it over to the yellow side and then open the back tab like this. And you'll see a full access to the sample stage where you're going to insert your specimen. Your specimen has a cover slip. Make sure that the cover slip side is down like this so that your sample is very close to the lens. I'm going to open the tab so that you see the arrows, and the arrows point to where you're inserting the slide. I'm going to insert the lower side, and then I'm going to flip over the yellow tab again, and you'll see the arrows, which points where you insert the other end of the slide, like this. And once they're securely in place, you're going to align your specimen with the lens, like this. I'm going to put the magnet together, and then face the blue side. To capture images on your phone with Foldscope, I'm going to use a ring sticker. So take another ring sticker and make sure you're peeling off the actual double-sided sticker like this. And you might have to really nudge it so that the sticky on this side. Now, put the sticker around the camera like this and peel off the top layer so that now the sticker is around. Make sure you're facing the black side and put the magnet coupler so that the center hole is aligned with the camera like this, and just press it so that it's securely in place. Now let's mount your Foldscope onto your phone. After you've attached the magnet coupler onto your phone, all you have to do is attach the Foldscope like this. Then now you can move around easily and then focus your image using the focus ramp. And take pictures whenever you see something very beautiful and interesting. So look. Use a digital zoom like this and take even closer look.
Experiment 3 requires you to prepare the pond water specimen ahead of time. So what you're going to have is a lyophilized pond water specimen. It's in a little packet in your kit, and you'll need to add that to some sterilized water. And that has to incubate at room temperature for 2 weeks before you use it. So your kit is going to have a little note on the lid notifying you of this when you pick it up. When you first get your kit, you're going to want to go ahead and set up the pond water specimen so that they have plenty of time to incubate before you get to Lab 3. You're also going to be using 2 pre-made slides. So these are slides that were prepared by me and Dr. Bhattacharya in the lab before you got your kit. You're also going to have a clean glass slide to use for this lab. You're going to be making 2 paper slides. So you're going to make 1 glass slide with the pond water specimen, and you'll use one of those clear stickers that are in the Foldscope kit as a cover slip for that. You'll use one of the paper slides to make a pond water slide. Then you're going to have 2 glass slides that are already prepared that you're just going to view. And you're also going to be making a wet mount with the paper slides with using a piece of your hair. So that is a grand total of 5 slides that you need to view for your experiment 3 procedures. You're making 3 slides, you're viewing 5 altogether. As for your pond water specimen, here are a lot of examples of what you might find in there. These guys over here on the left are all swimmers. So if you see things swimming around, you see things moving around, it's probably these guys. Amoebas move using pseudopods, false feet. Most of these others are using cilia or flagella. The paramecium here, these guys are just covered in cilia and they move very, very fast. These guys that have like one flagella at one end, they move a bit slower and they whip around, like they whip back and forth. Things like this and this guy here, they kind of swim around in circles. So you could see any or all of these things. It really just kind of depends. Your Spirogyra are pretty cool. They're rather large, eukaryotic, multicellular, and they are green because of all of these chloroplasts that are inside. Algae, you're going to see probably a lot of algae. It's greenish or brownish. In fact, algae are a good place to start looking for swimmers if you're looking for swimmers, because these guys like to eat the algae. So if you find a big clump of algae first, you kind of focus in around the algae, you may very well find some of these guys. So as far as safety, there really isn't much in this lab that is hazardous, but you do need to know general microscope care. And as far as what you guys are doing specifically, you want to be careful not to crack your glass slides. Be careful with your Foldscope, especially when you're assembling it. You don't want to tear it. Take care not to drop or crack the lens or the magnetic coupler that comes with it. And when you're disposing of your slides, you want to make sure that you're either putting it in a homemade sharps container that has hard plastic sides, or you're wrapping it in cardboard or some kind of bubble wrap or something that's insulated so that it won't break in your trash can. Some of the observations, they're pretty straightforward. Were you able to identify all the parts of the scope? Do you know how to use the parts of the scope? Were you able to observe specimen and things using the tutorials and simulations? Were you able to make your wet mount slides correctly? Were you successful in viewing your wet mount slides and the prepared slides? Do you know why a cover slip is necessary when you're using a wet mount? That should become pretty clear to you when you're actually trying to view the wet mount slides. So I hope you really have fun with Lab 3. The Foldscopes to me are very interesting, and they're not as good as the scopes we use in lab, obviously, but we're going to be looking at things that are on a larger scale as much as possible. When we get to some of the smaller things in labs 4 and 5, you're going to see even more online simulations and stuff. But for Lab 3, you should be able to see all of these specimen. You should be able to focus them. Spend some time working with your Foldscope, get used to manipulating the slides and things like that, and have fun with it. Okay, well, thank you guys for watching, and don't forget to do the reading and check the description for more videos related to these topics, and leave your questions for me in the comments below.